I do. Hi. Welcome.
Thank you. Right.
Nothing's going.
Don't rip me to pieces over my very simplistic model of gene
No,
regulation.
no. I don't let people rest.
Okay.
I leave that to the others.
Okay.
How much you need? Water.
I've got water. Got
There's
everything.
time to run to the bathroom if you want to.
I'm good.
You're okay. Somehow when I'm speaking, I always the last one. I'm like, I'm going to go to the bathroom. As you can tell, this lecture hall is usually used for medical school lectures. So ergo skeleton and a massive skull there.
I don't think I'll need that.
You don't need it.
Hope not.
He's going to
You
show
never
us where
know.
the brain is.
Never know.
John Assad is right now in Italy, but he'll be watching your talk it'll be recorded for him.
Sebastian said that would be the case. So I asked him if I could actually have a copy of it
Oh, nice.
because I just find it nice, well, embarrassing and useful.
That's a really good idea.
You know, to see what I'm doing well, what I'm
Great
doing
idea.
less well.
Good. They're usually a pretty responsive audience. Like, they kind of get into it.
Okay.
They're not going to ask a lot of questions during the talk. Someone might, and it's a good idea to take that question, just because it's probably going to be some point that needs to be clarified. People will hold the question to the end. But it's a fun group because, like, it's a pretty tightly knit department. They get together for several seminars a week, usually, and you can kind of feel the vibe in the crowd where people are like, yeah. It's fun to speak to the crowd.
How tight am I going to be kept to time?
Pardon? Yeah, don't worry too much. I mean, I think generally I'll give a really short introduction, then you would probably talk for about 50 minutes. If it's 55 or even close to 60, don't worry about it. No one's going to get up and leave. There's one or two people who do just have some appointments.
Sure.
But, and then we'll let the question period go on. We can let it go on a little bit longer if there are a lot of questions and your talk is on the short side. But if your talk ends up running a little long, we'll just keep the questions a little bit shorter so people don't get restless. But it's pretty flexible, don't worry about it. There's not like a hard thing that everybody needs to run off to. I think most people would probably expect not to leave the auditorium until 12:45. Don't worry about it. And when it's done, I'll let you call on people to ask questions.
Sure.
And I'll just shut things down if people will not shut up. So don't worry about that.
I'm keeping my voice down because I'm mic'd up.
Oh, I see.
Oop. I got attention.
Your turn.
It is on.
Preset panel is right here.
Yeah. Right, so I think it's 3, right? Move right.
Much brighter. Did you want it? You want 4?
All right. Hello. Glad to see so many people here. So if you have spent sort of a lot of time, as I have, listening to Masterpiece Theater or Downton Abbey and things like that, then you might pride yourself in your ability to tell a Yorkshire accent from a Cornish accent. And you'll be quite puzzled and intrigued when you listen to Timothy O'Leary begin to talk, because he quite clearly has a delightful regional accent of the British Isles, which is completely unplaceable. And so I asked him about this morning, and it turns out that he is, quite broadly speaking, a resident of the Celtic fringe of Britain. His family is both Welsh and Irish, but he spent time growing up in Scotland, ergo the Celtic fringe aspect. These are known as wild and ungovernable regions of the British Isles, so we'll see how that plays out. But once he starts talking, you don't pay attention to the accent anymore, because he's actually a really interesting person. He studied math as an undergraduate at the University of Warwick, and then quite unusually went on to do a PhD in cellular neurophysiology, working at the University of Edinburgh with David Wiley. So his PhD thesis work concerned the single-channel properties of NMDA receptors, and also the homeostasis of intrinsic excitability in hippocampal pyramidal neurons in dissociated culture. And that led to a general interest in homeostasis and led him to do a postdoc in Eve Marder's lab at Brandeis. And I think he'll tell you about that work today, but he's focused on using modeling to understand how mechanisms of gene expression might govern the pathways that neurons use to produce homeostasis. And I think we're really pleased to bring Dr. O'Leary here to give us a talk, in part because we'd like to bring more theory into the department, frankly, but also because he trained as a synaptic physiologist, and so he has a, I think, really clear idea for what the texture of real data looks like. So welcome.
Okay, thank you all for being here, and a special thank you to the search committee for inviting me. This is, as well as being truly terrifying, this is a great honor. So I'm pleased to be here. So I'll just get right to it, and I'll start with something that I think we can all agree upon, which is that the nervous system is made from neurons, and that neurons are electrically excitable. And here's two neurons. If you have an action potential in a presynaptic neuron, this may sometimes, if it's synaptically connected, cause an action potential in a postsynaptic neuron. Now, this humble-looking mechanism is widely believed to underlie every thought, belief, perception, action that we have. So it's crucial that the excitable properties of neurons are regulated somehow. Now, neurons don't just sit there and fire action potentials in stereotyped ways. There are many types of neurons, all with different stereotyped electrophysiological signatures. So it's very important that these different neuron types also maintain their electrical properties. Now, what determines electrical properties? If we zoom in onto the membrane of a neuron, we'll see that it's teeming with ion channels, of course. And ion channels are very nice because we can understand them on some level. We can build abstract models of them, and we can reconstitute a circuit that in some way behaves like the neuronal membrane. And these are the only sets of equations. Actually, that's not true. These are probably the only sets of equations I'm going to put up, and they're important because this really gives a flavor of all of the modeling work that I do in this talk. The top equation is just the membrane equation. This just says that the way that membrane potential dynamics operate is a sum of all of the membrane currents, which we all know. And the other important part of this is in the blue box, and this just says that ion channels or membrane currents have internal dynamics as well, and this interesting, rich internal dynamics is what gives neurons their flavor. And of course, this goes back a long way. This goes back to the 1950s, the seminal work of Hodgkin and Huxley, where they showed for the first time that you could get a quantitative description of electrical excitability in nerve cells. And since then, we've extended the repertoire of the types of phenomena we can account for. And this is wonderful, right? Because what it means is that we have a roadmap for solving the brain. All we need to do measure physiological parameters in neurons and circuits. We can build real mechanistic models of these neurons and circuits, make and test predictions, and then define new experiments, and we go round and round this loop until eventually we understand how the brain works. Now, the problem with this is that it normally looks like this, at least in my experience. We can't measure everything. So we often have to make some kinds of assumptions, and often it pays well to ignore the contradictory data. A lot of people end up here. You can spend your life here, and you either leave feeling somewhat depressed, or you move on to a problem in applied mathematics. So why is this? Is it just that computational neuroscientists like myself are incompetent, or is there actually something deeper here? And does this actually mean that we need to relax our idea of what a model actually means in biology generally? Well, let's take a model. Here's a model of a pacemaker neuron, and it has lots of conductances underlying this nice bursting behavior. And the nice thing about a model is we can do a thought experiment. So we can say, let's pretend this is a real neuron and imagine that the conductance expression varies by some modest amount, 20%. In biological terms, that's not much. Okay, well, this could happen. Or any of these things. So if it's really important for the organism that these properties are maintained and a 20% variation can completely destroy them, we already have a problem. On the other hand, 20% variation may not destroy the properties entirely, and this relationship is, in general, quite complicated. So this sets up the general question, how variable are conductances biologically? And to address this question, of course, we need a well-defined neural circuit. Where do we get one of those? Well, we just go down the fishmonger and get a crab. So here's a crab, and inside the crab, there is the stomatogastric nervous system, and there's a small rhythm-generating circuit here called the pyloric circuit, and all this circuit does is it generates this stable triphasic rhythm for the entire duration of the animal's lifetime. But the wonderful thing about this is that each crab only ever has one LP neuron, for example. So you actually get to ask the question in two different individuals, if we find the same neuron, does it have the same underlying conductance properties? And in the knowledge that the fact that it has the same, by and large, intrinsic properties. And of course, I wouldn't be here talking to you about this if this was not the case. So here are two different LP cells with very similar membrane potential activity. The underlying currents are quite different. And across a population of different LP cells in animals, we see this enormous range relative to a 20% range anyway. So this seems like a serious problem. Now, you might say, well, okay, it's hard to measure membrane conductances. This could be experimental error. So we need some independent way of validating this result. And several years ago, some people in Eve's lab did some single-cell real-time PCR on these neurons, and they found, first of all, that the RNA copy number correlates very nicely with the overall conductance expression. The other thing they found, reassuringly, or perhaps unreassuringly, is a large variation in the expression levels of all these different ionic conductances. Now, the advantage of single-cell PCR is that we can look at multiple different ion channel genes in the same cell at once. And when you do this, you see this. So there are striking correlations between the different ion channel genes in each cell type. Moreover, each of these different cell types, and their activity is shown here, seem to have a unique signature in this correlation pattern. So this starts to make sense. It's not just the case that the conductances are randomly distributed. There's some sort of pattern here. The next obvious question is this pattern actually essential for the membrane potential activity of these cells? One way to address this is to build a detailed computational model, but to be agnostic about what the parameters of this model are. And to just say, we want the output of this model to look like this, but it can do so with any kind of distribution of intrinsic conductances and receptors and so on. So we build these random models. Here's the model cells, and we only select those that are functionally close to the biology. And in a large search of nearly 1 million models, you find a small fraction of them are actually functional. And now you get to ask, are there any correlations between the parameters of these functional cells? And there's none evident almost whatsoever. So here is an A-type potassium current, and here's a delayed rectifier current, and you can see that the solution is distributed everywhere, even though there are tight correlations in the biological case. So what on earth is going on? This is the summary so far. Models let us interrogate this universe of all possible solutions. And we find that for any given phenotype in a cell, there are solutions everywhere. In the biology, it's not the case that we ever have unique solutions. There's variability in the different solutions, but there's some kind of pattern. So this sets up the obvious question, where do these correlations come from? And this is related to the more general question of how do neurons and circuits actually find their parameters in the first place. So the first thing to remember is that these parameters are not fixed. The receptors and ion channels in the membrane are turned over continually, and for a long time it's been hypothesized that to keep this system stable, you need some sort of homeostatic feedback control. And the next part, I'm going to describe some simple experiments that I did in a completely different system. This is not in the crab. This is in hippocampal neurons that are grown in a dissociated culture like this. And the way that we assay the membrane potential properties is with whole cell patch clamp recordings. And the advantage of this system is that you can manipulate it quite easily. So here is a recording of a cell where we've just washed in 15 millimolar KCl, and we can depolarize the cell and raise its activity. And we can do this chronically. So here's a control cell, and we have a simple assay of its excitability by injecting depolarizing current, and we reach some current threshold where we elicit action potentials. And now we take a cell that's been grown for a week in these high potassium conditions where it's been depolarized continuously, and recorded under the same conditions, we see a dramatic increase in the amount of current that's required to elicit action potentials. This is what summary data look like. So here are the control cells. This is the amount of current and the number of spikes you get for a fixed current pulse. And this FI curve here, you see shifts dramatically and increasingly to the right as you increase the extent to which these neurons are depolarized. So this really starts to look like some sort of homeostatic mechanism. The cells are becoming less excitable as they're more depolarized. Another important aspect of this is that the effect is reversible. So here are the control cells again. Here's the FI curve for cells that have been depolarized for a week. And then if they're just returned to normal conditions, the FI curve jumps back in almost 2 days. And here's the profile of the threshold for initiating action potentials as a function of time. And this is exactly the type of profile you'd expect if there was some sort of feedback control mechanism tracking average activity and maintaining excitability of the cell in order to achieve some kind of set point. Okay. So how do the neurons actually sense activity? Well, you all know that when you depolarize neurons, many things happen, and one of the important things that happens is you get a large surge of calcium influx, and we see that here in this calcium imaging experiment. We can also manipulate this calcium influx. So here we have 15 millimolar KCl, and then when we reach some sort of steady state or plateau in the calcium influx, we can attenuate this quite substantially by applying nifedipine. And this gave a route to manipulate this perturbation in vitro. Here's a control recording, again, from a cell that's a week old. Here's a cell that's been depolarized for 60 hours, and it's become less excitable. Here's a cell that's been depolarized for 60 hours, but has had calcium channels blocked with nifedipine, and it's almost as though the cell hasn't noticed this depolarizing stimulus. And this is what the summary data look like, and they're actually quite striking. This is the depolarized condition where we get this rightward shift, and then the nifedipine condition is almost indistinguishable from the control condition. This is actually quite remarkable because underlying this change, a number of currents alter their expression. Sodium currents decrease, various calcium currents decrease, and various potassium currents increase. So this simple-looking calcium signal is causing the coordinated response of the expression of multiple ion channel subtypes in these cells. Okay, so I hope I've convinced you that among the various possibilities for this feedback control, we have calcium as a key feedback signal. Okay, so this gave the inspiration for some modeling work. We can go back to our model cell. It has calcium dynamics, and we can use these calcium dynamics to feed back and control the expression levels of all the conductances in the cell. Now, this seems like a terrible idea because, as I pointed out, these cells are extremely sensitive to even modest changes in the expression levels of conductances. So there's no reason on the face of it to expect that if we just plug this back into here, that we don't get some horrible positive feedback loop and everything explodes or falls apart. But I hope to convince you that actually it's not as bad as that and that this system is actually quite easy to understand. So here's the setup. We have this model neuron with some calcium dynamics. We feed this back through some control rule, and the control rule regulates the expression levels of the membrane channels and receptors. What's this control rule? That's key. Well, in this case, we don't know much about the innards and the workings of how cells regulate their conductances, and that story is a work in progress. So I took as my starting point the simplest possible control rule I could think of, central dogma. So calcium, in a very naive sense, which I hope won't upset people like Michael Greenberg, it turns on and turns off stuff. In this case, it turns on and turns off the expression of ion channel RNAs in this model. And of course, these RNAs are turned over and degraded at some constitutive rate. And then, of course, we get conductances in the membrane just in rough proportion to the amount of RNA that's around. And these conductances are turned over. And when you boil together this very simple model and you make a sort of minimal mathematical model of it, this is the second equation and possibly the only one, you end up with this. You end up with a few free parameters. You have this notion of a calcium target, which is just like an equilibrium that some transcription factor likes to have in the nucleus. And you have the rate of change of the RNA with respect to the difference from this target, the difference in calcium concentration from this target. Now, the rate at which these RNAs are expressed, that's a free parameter. And I'm just going to play with this parameter and just show you what happens to begin with. So what we've done here is we've taken a cell, we've put some conductances in it, we've put some RNAs in it, they're randomly distributed, so it's disorganized, and the cell is not excitable, it's just sitting here at rest. Now we set this model running, and this is what happens. The conductances all change their expression and settle down to a steady state, and the cell then acquires this nice rhythmic bursting behavior. Now, we chose these initial conditions at random, so the next question to ask is what happens if we choose different initial conditions? Well, if we keep running this model over and over again, this is what we see. Here's the model as it develops, and here's a population of cells that have been grown with the same regulatory control rule, but from different initial conditions. And what we see is that all of them have this nice bursting phenotype, but they're all somewhat unique in character. And here you can see that the expression levels of all these conductances are somewhat different between each of the cells. It's squashed here because we're on a log scale. So what I'm going to show you now is the distribution of the conductances in an easier way. And I'm going to plot the pairwise correlations between them. And this is what you see. Straight away, you see these striking correlations between the expression levels of the conductances, and you see that they can vary several fold, in spite of the fact that the properties of these neurons are all largely similar. Okay, well, this is nice. Another nice feature of it is that the equations are relatively simple, and we can come up with an expression, an explicit expression for the slope of this correlation. Why do we care about this? Well, there's a few reasons. One is, if we believe this model is in any way approximately correct, the readout of the steady state expression levels of these ion channels gives us a clue as to the dynamics of the underlying control system. The other reason this is nice is that we can actually now manipulate this correlation by manipulating the parameters of our control equations. So we can generate populations of cells with different correlations that have grown from the same random initial conditions. And this is what happens when we do this. Here are 5 different populations of cells, all grown from different random initial conditions. All grown from the same distribution of random initial conditions, but with different regulation rates. So they all have their own unique correlation patterns. And when we look at the intrinsic properties of these cells, this is what we see. We've got cell types. So we've got type 1 regular spiking neuron with an FI curve that looks like this, a type 2 regular spiking neuron with this sharp threshold, a rebound bursting cell, bursting pacemaker cell, tonic pacemaker. All of these different cells are found in these clouds shown here. So this gives some clues to, in the model universe at least, what a cell type is. A cell type is not determined necessarily by the absolute expression level of any one particular gene. If we looked at this calcium channel here as a cell type marker, it's going to do a pretty poor job. But what we need, when we look at the joint expression or the correlation between the expression of these different ion channels, of course, then we see interesting patterns and we can identify cell types. Okay. This hopefully is reminiscent then of the biological data where we saw that indeed different cell types have these different characteristic expression patterns. Okay. So this is nice. We started with this mess where the model or models are not particularly well constrained by the intrinsic properties because they have many, many different ion channels and many, many different ways of getting those properties. And the biology is somewhat variable but constrained. And the answer, at least in part to 2 of these questions, seems to be that feedback control is the missing link. And that's very nice, that's an attractive idea. But at this stage, some people say, why? What is it about the feedback control that's giving these correlations? You could just take my word for it. You could just believe that the maths works out that way. I don't want to leave you in that state. I want to give you an intuitive picture of why you get correlations with this very simple-minded feedback control rule. And in order to do this, I'm going to go back to a very unrealistic universe where we just have this simple toy model cell. It only has 3 conductances. They're essentially leak conductances. They just depolarize or hyperpolarize the membrane. And inside the cell, we have fictitious calcium influx, which is just essentially membrane depolarization. So it's the simplest possible thing you can think of. And all we want to do now is study this feedback control rule in this context. So here we go. Again, we have this calcium target, and we start this cell from somewhat random initial conditions, and each of these ion channels has its own characteristic time constant that couples it to this error signal that's encoded in calcium. And you can see that the model settles down, and you can probably guess that it's important to choose the direction in which these ion channels move in an appropriate way. So if the cell's too hyperpolarized, it should upregulate its depolarizing conductances, and so on. Okay? So an easier way to understand what's going on is to plot this thing in 3D. Here's the 3 different conductances. Here's the initial conditions. And here's the cell as it settles down to its final state. And the evolution of this system through this space is just the derivative of the conductance with respect to time. You don't need to worry about that. It's just there for people who like to think that way. So if we, again, start from different random initial conditions, we settle down to our steady state, but this time with somewhat different conductance values. If we change the characteristic time constants at which each of these conductances are expressed, we move this trajectory. And then we end up in a different point in parameter space at steady state. So here we just scaled these expression rates. We can even do something that seems like a terrible idea and actually make one of the conductances go in the wrong way. So this is an inward conductance that should actually upregulate because the neuron starts out too hyperpolarized, but instead it does a stupid thing and downregulates. But because there's redundancy in this system and there's 2 other conductances that can do the job, the system is still actually stable and it converges to its target. Okay? So we can run this over and over again from this cloud of initial conditions, each with these different characteristic regulation parameters, and we build up these different clouds of steady state conductances. So these are like the different cell types, all growing from this random initial state. And when we do this long enough, we see two things. We see that all of these solutions, of course, will land on some space. We have one constraint. We've got this one sort of target membrane potential, and we've got three conductances with which to achieve that potential. So this defines a two-dimensional surface. So all of the steady-state solutions need to sit on this surface somewhere. But because the regulation rates that define the trajectory of how these cells move through this space are different, they impact this solution space in different ways. And then straight away, I hope you can see that if you looked at the pairwise expression levels at steady state, you'd see that all of these things have correlations. And these correlations in some way depend on what the relative expression rates are. The other thing that comes out of this picture that's very important is that it's not so hard to build a cell that's stable. If we start out on one side of this solution space, we just need to choose a trajectory that takes us towards it. And across all of the possible choices of trajectory there are, 50% of them will take us in the right direction. So there's actually a huge amount of flexibility in the system because we're only using a single control variable and we just need to satisfy a single target. Okay. So I hope that somewhat clears up the intuition for why this model works. But I'd still like to summarize some main points here. So correlations in conductance arise automatically in self-tuning homeostatic models, and the correlations themselves depend on the relative rates of the regulation mechanism. Simple feedback mechanisms tolerate a substantial sloppiness in how individual conductances are regulated by activity. We can actually manipulate things so that they move in a somewhat anti-homeostatic way and the model remains stable. And this is important because it means that we can generate different correlation patterns and different cell types with, by and large, the same regulatory mechanism. Finally, consistent cell types, and I'm somewhat repeating myself here, consistent cell types can be nonetheless defined by cell type-specific regulation rates for different ion channels. This is compatible, of course, with cell-to-cell variability. Okay. So at this stage, everything seems very nice. Things are starting to make sense. But it all seems a little bit easy. Surely things must go wrong when we just make this simple-minded model and feed back the expression levels of all of these conductances using this simple signal. Okay, well, here's one of these model cells at steady state, and it's got this nice rhythmic bursting activity. Here are the calcium dynamics. You see these nice calcium transients. And then the steady state, or the average calcium, sorry, not the steady state, the average calcium has, in this case, of course, hit its target. Okay, so this cell is happy and it's settled down. So you can imagine this is a wild-type cell. It's just sitting there doing nothing, and we haven't manipulated it at all. And of course, the first thing that a neuroscience does when it sees something that's wild-type is knock something out. So here, we've taken this cell and we've acutely deleted a conductance from the membrane, a calcium channel in this case. Not surprisingly, the cells become silent, and the calcium the average calcium has dropped beneath its target value. And then when we let this simulation run further till it reaches steady state, the cell now recovers its target, and then also, importantly, it recovers its nice bursting properties. So what this says is that this simple-minded feedback regulation rule can give rise to an emergent homeostatic behavior, that this neuron sort of wants to burst. It wants to fire these bursts of action potentials. And in some cases, when we stop it doing that, it finds a way to do it by other means. Because this model generates inherently variable populations, we see, so here we have two different cells grown from the same random initial conditions with the same rules. When we delete this ion channel gene, in this case, we don't silence the cell. And this is something that you see in biology. There's variable effects, even though perhaps you might have a clonal population of cells or of an animal, or whatever. So this model automatically gives you, in some cases, some cryptic variability and some strong sensitivity in some cases and lack of sensitivity in others. And then in this case as well, when the model reaches its steady state, it preserves this bursting behavior. However, if we delete now a different type of calcium channel, again, we silence the cell, but now when it recovers to its steady state as far as the average calcium is concerned, the overall intrinsic properties have completely changed. So the control system here is quite happy. It thinks everything's fine. But this calcium channel here was clearly crucial for some aspect of the intrinsic properties of the cells, but it's not there anymore. So the remaining channels have done the job in its place, but they're unable to bring the cell back to this bursting behavior. So we think that this provides quite a compelling model of aberrant compensation. And normally when we think about disease, we think about loss of homeostasis or some malfunction of a regulatory mechanism. But this is an example where actually the regulatory mechanism itself has in some way caused a problem. Of course, the initial insult has as well. But it can get even worse than this again, as I'll show you. So here now is a different cell type. This is a tonic spiking cell. We delete a conductance, it stays a tonic spiking cell, and then when it reaches steady state, it's still a tonic spiking cell, so this is fine. If we delete this other conductance now, however, again, the cell remains a tonic spiking cell, and this might not be catastrophic for the circuit or for the animal. But now when the homeostatic mechanism recovers to its target calcium level, the cell becomes completely silent. So this is an example where the regulatory mechanism itself has actually caused the problem. And one thing that I wonder more generally is that in many kinds of diseases, the nervous system, and outside the nervous system, just in general, we get this kind of thing happening. Okay. So in summary, the basic reason for this aberrant compensation, as it were, is that all of these different activity patterns can have the same average calcium value. And in this simple regulatory control model, it's only the average calcium that this thing ends up caring about, okay? So what we've done is taken several different phenotypes, squashed them with a very crude signal, and then used this to regulate all of the things that are important for generating these phenotypes in the first place. In some cases, this works. In other cases, it doesn't. And you might say, well, this is actually kind of a crappy model, but there's an advantage to this as well. If we're using this one variable, then, as I explained before, it's somewhat easy to get to the target for this variable because there are relatively few constraints. There's also other things that we can do that are interesting, and I'm going to get to those right now. But before I do that, I'm just going to talk very briefly about an interest that we had in the lab and a use for this model. So can this simple feedback model, in spite of its simplicity, be tuned to cope with acute global perturbations? So what do I mean by that? Well, crabs live in the sea, and the sea can vary in temperature quite substantially. Why is this a problem? Well, crabs, of course, have nervous systems, and nervous systems need to carefully tune their properties to maintain activity. And if we measure the underlying currents that are important for maintaining this activity, in this case, recording is done to show the IA, so this is A-type potassium conductance activation rate, you see straight away that this has quite a steep temperature dependence. Temperature dependence defined by the Q10, so this is the fold change in a property per 10 degrees Celsius, okay? And this is quite a common value for a Q10 of about 3. Importantly, other properties of this membrane current, in this case, the inactivation rate, also has a very steep temperature dependence, but it has a Q10 of nearly 4. So here we have a situation that within the same ion channel type, the activation and the inactivation variables can get detuned as the animal experiences a temperature change. And this gets worse and worse again as you consider the different Q10s that other ion channel types have in the same circuit. So just to illustrate this point, here's a simple model neuron at 10 degrees Celsius. And we've been agnostic about what the Q10s are, so we've just randomly distributed the Q10s of all the ion channels in some physiological range. And now we're going to temperature ramp this neuron. And even a modest change in temperature, certainly not as big as the animal might experience, critically disrupts the activity of this neuron. So when Q10s are not matched, physiological properties are generically destabilized by temperature perturbations. And before you say, well, I don't care about this, these are crabs, I'm interested in mammals or warm-blooded things, I'd just like to remind you that not all of our nervous system stays at 37 degrees Celsius. Moreover, sometimes the brain itself doesn't remain at body temperature. These are some experiments from several years ago, the Morris Watermaze experiment, where Edvard Moser and others put a thermocouple into the hippocampus and measured the temperature of the hippocampus in this poor rat when it was dropped into cold water to perform the Morris Watermaze task, then warm water, then actually pretty hot water. And what you see is that the temperature varies by 10 degrees Celsius. And this variation in temperature is accompanied by quite striking and scary changes in the physiological properties measured by a tetrode. So the EPSP slope in this region drops to almost nothing, and the spike amplitude and spike latency is completely messed up. Now, the important thing to note here is that this animal was fine. It was able to complete the task. It was able to navigate. But its brain had changed temperature, and all of the circuit properties had changed quite drastically. So it's clear that this is quite relevant to a favorite model organism, and probably relevant to humans on some level. And even if it wasn't, understanding why this doesn't happen in humans is the flip side to understanding how it can happen in other animals. Okay, so back to our crab. The remarkable thing about the pyloric circuit is that if you change the temperature by nearly 20 degrees acutely, so this is within a few minutes, then of course the cycle period speeds up. But when you look at the phase relationships between the firing of these different motor neurons, they're almost perfectly preserved. And if you remember back to the simple model that I showed you, you wouldn't expect this if the ion channel Q10s are mismatched, and indeed they are. Okay, so how can this work? Well, again, we return to a model and ask the simple question, if we just have these model cells and a simple property that the cells, that's important in these cells, in this case the duty cycle, this is the duration of time spent firing relative to the interburst interval, we're going to assume that this property is important, and we just want to preserve this. And we're going to randomly assign Q10s in this population of cells. Okay. Now, the first thing we need to do here is some math. I'm sorry about that, but it's not too bad. P here is the property that we want to preserve. In this case, it's duty cycle. And all we're saying is that we want the derivative of P to be roughly 0 with respect to temperature. That's just another way of saying that this thing is temperature robust. And then if we use the chain rule, and X here represents gating variables for all the different ionic currents in the cell, we see that this derivative is a sum of how the property changes with respect to the gating variables times how the gating variables change with respect to temperature. Now, this latter term is just the Q10 relationship, or derivative of it, okay? So this is the thing that's unmatched. This term just says, how does my duty cycle change as I change the amount of A-type potassium current I have in the cell, etc. This can be either positive or negative, okay? And these are a bunch of random values. And all we need to do is to get this thing to sum to 0. Well, you can see straight away there's going to be lots of ways of doing this if there are lots of conductances in the cell. So you do some horrible maths, and at the end of it all, this comes down to this local constraint, which just says my vector of conductances, so this is the vector representing the expression level of all the conductances, needs to be orthogonal to 2 other vectors. We have 8 conductances. We're in an 8-dimensional space. We need to be orthogonal to 2 vectors. So we have 6 dimensions left. This is quite an easy problem to solve. And it's even easier in this regulatory control model because we know that if we have a cell here that's temperature robust, then any other cell that's generated with the same regulatory control rule will be on this same vector. So it too will be temperature robust. And indeed, that's the case. So here's a model where we've tuned the regulation parameters to make the cell temperature robust. And this is an acute temperature perturbation where it goes from 5 degrees Celsius to 25, and we can more or less perfectly preserve duty cycle, even though, again, the rate of these action potential bursts changes significantly. And we're not cheating. The Q10s that reside to this cell are not matched, and they're substantially different for a number of important gating variables. And in summary, this is what it looks like. We can go from this case with no regulation to a regulated case and preserve certain properties of the neural activity. Okay, so that's very nice. In this last part of the talk, unless there are sort of burning clarification questions that I could take right now, I'm going to talk about some future work that I'd like to do. So does anyone have, oh, this question.
You said with regulatory constraint. Was that regulatory constraint the old calcium regulatory constraint, or was it your new, you have to maintain your duty cycle constraint?
We were using the calcium signal as the feedback. And the basic idea is when you use the calcium signal as the feedback, you get these correlation patterns. Now what we need to do is get the correlation pattern that satisfies these other constraints. So the argument is this is something that would have to happen evolutionarily, obviously, right? So the cells that are grown with this temperature robust rule are inherently temperature robust. They don't need to adjust their conductances to compensate for temperature changes. Okay. Well, one of the future directions or future sets of questions I want to pursue is this idea of regulatory rules. How can regulatory dynamics be inferred, to begin with, from gene expression data or physiological measurements in real cells? And just to illustrate what I mean by that, here is an example in the crab STG where we had these different cell types and they had these different correlation patterns between the RNA expression levels. And what I said was that this simple dumb model can be solved, and we can get an expression for what the parameters of this model are in terms of the steady state expression levels of these RNAs. Now, of course, this is a nice case. This is simple linear correlations, and this is a very simple-minded model. But more generally, we might have more complicated regulatory control schemes, and this will give us different steady-state distributions of conductances. So understanding generally what types of feedback control rules are plausible and how they shape the steady-state correlation patterns of conductances and receptors and so on is a really important problem. Okay. Beyond that, what are the special problems that neurons face in regulating their electrical properties? Most of what I've been talking here about the neurons, in a sense, in isolation, or it's subject to sort of minor perturbations of a change in leak or deletion of a channel or so on. But neurons are obviously far more complicated than just single compartments. They have long neurites, and there are various constraints that might exist on the regulatory mechanism to ensure that the neurite retains its appropriate level of excitability in spite of transport delays of RNA, in spite of noise in gene expression, and so on. So there are a lot more details to be fleshed out in these simple regulatory models, and a lot more basic cell biology problems that can be addressed. And what can broad brushstroke models tell us about the etiology of pathological states in the nervous system? So this is, I think, a real-life example of a system where we have pathological compensation. So this is a mouse model of Dravet syndrome. So Dravet syndrome is a form of epilepsy, and the gene that's affected in Dravet syndrome is SCN1A. This encodes a sodium channel subunit. And in this mouse, we have a het for the knockout. And what we find is in that these parvalbumin positive interneurons in the cortex, these cells are markedly less excitable. And this kind of makes sense from the point of view of epileptogenesis. We have a whole population of interneurons that have lost a degree of their excitability. But the interesting part of this is that this hasn't just arisen because these things have smaller sodium currents per se. In fact, what's happened in the het is there's been partial compensation by this NAV 1.3. But the biophysical properties of this sodium current are not appropriate to generate the fast spiking behavior of these cells. So here what we see is that the cells have somehow sensed that something's missing, and the feedback control mechanism has responded by upregulating NAV 1.3. But this has not resulted in compensation of the properties of the cells, and in fact, it's actually resulted in pathophysiology. Okay. Next set of future questions is the question of neuromodulation. So feedback regulation mechanisms need to be compatible with variability and ensure stereotype circuit properties, but they also need to allow neurons and networks to dynamically reconfigure. How? What I mean by this is that we have this issue of reconciling variability with stable function. But this relies on quite a specific distribution of conductances. Now, modulators change the dynamics and the conductance of all these different conductances. So how can you have reliable modulation when you have these variable conductance expression levels? Here's a real-life example of neuromodulation that's very important. This is ongoing activity in the cortex during slow-wave sleep, and you see that it's organized into these bursts. And then during wake and REM sleep, we see this more sporadic pattern of activity. And this, of course, is not just present in the cortex, it's present in the thalamus, and this is a classically studied system for modulation, because thalamic neurons are able to switch between this burst mode and tonic spiking mode via the action of various neuromodulators. And people have thought for a long time that this is a serious problem for regulatory control. If there's some sort of feedback mechanism in these cells that says, okay, I want to be a bursting cell, and I need to maintain some sort of average activity that corresponds to bursting. If I also need to now be able to switch for a long period of time to this tonic state, how can I do this without the compensatory mechanism interfering with this switch? And if you remember, I said that this very simple regulatory control model uses, in a sense, a dumb readout of what activity is. It just takes average calcium. And average calcium, of course, can correspond to a number of different overall physiological properties. So in this model, if we were able to switch the conductances by some value that takes us from this bursting state to this tonic spiking state, what we'd do is change the activity of the neuron without actually affecting the average value of the calcium. So this is one way in which you can make chronic or acute neuromodulation actually compatible with regulatory control. It's not always going to work lickety-split. There's going to be some cases where there could be variability in the population of neurons where we actually don't hit the calcium target bang on when we modulate. And that's going to be interesting, because there we're going to get a pathological state induced by chronic or acute modulation. And to some extent, we've solved the baby case. Here's an example from some work that's ongoing, where we have a population of these tonic spiking cells, population of bursting cells. They all have variable parameters, and if we just change the maximal conductance of these three conductances by some percentage that's approximately correct, we can switch these cells from bursting to spiking without messing with the homeostatic feedback mechanism. Okay. So this leads to a more general question, again, of what kinds of feedback control mechanisms might regulate the component that underlie modulation itself. So here we had feedback control regulating all of the ion channels that get modulated. But if we're going to have stable modulation on top of variability and on top of feedback control, presumably we need to somehow tune the action of these modulators. And one question I'm very interested in, just from a conceptual level, is what kind of feedback control mechanisms could underlie this kind of tuning? And when might modulation, as I mentioned, actually interfere with other regulation mechanisms in a pathological way? You can think of addiction, and you can think of a number of interesting pathological states that might actually arise from crosstalk between a homeostatic mechanism and a modulatory mechanism. Okay. So the final sort of theme questions that I want to pursue are trade-offs in the nervous system. So what are performance trade-offs in neural circuits? So what do I mean by that? Well, I told you about these neurons that we were able to make, these model neurons that are temperature robust. And these neurons do not a bad job. They keep their duty cycle maintained within about 5% of some nominal value. So if we think of 5% duty cycle maintenance over a 20-degree Celsius change as a measure of how well these cells perform, we might say, well, if these guys maintain their duty cycle within 5%, does this actually come at the cost of something else not being maintained? And indeed it does. So here's a population of cells. Here's the duty cycle robustness as assessed by the amount the duty cycle changes over a 20-degree Celsius change. And here's the robustness of the interburst period. Now, in our model universe, we weren't assuming that these cells compensated period, but this is another physiological property of the system that might, in other circumstances, be important. So here are the two extreme examples. Here's the best performer in terms of duty cycle, and you can see that its period changes quite drastically, and its duty cycle is nicely preserved when we stretch the traces. And here's the best period performer. It keeps almost perfect rhythm frequency over this 20-degree Celsius change, but you can straight away see that the duty cycle is not preserved. And the important thing about this collective set of data is that the so-called Pareto front, which is the set of all possible solutions where you do as well as you can on this thing without doing, and you do as well as you can on this thing at the same time, is concave. So what that means is that there's actually a hard trade-off, and that if we're doing well at this, it comes at the cost of doing well at that. And the only way to beat this hard trade-off is actually with a mixed population of solutions. So if we imagine a population of these two cells, and we have 50-50 mix right in the middle, then our performance for period and our performance for duty cycle robustness beats any one individual solution. So this is one example where variability actually might serve a purpose, not just between individuals, but possibly within the same organ. It might be the case that within the brain we have variable populations of cells because they're trying to cope with these contradictory trade-offs. And this is a general theoretical question that I'd like to explore, and obviously it'll be inspired by a lot of the ongoing work here in specific neural circuits. And the first thing to address is actually what are the performance trade-offs? And this isn't something that's terribly well fleshed out in neural circuits. And then beyond this, is there in fact any evidence that variability here might be exploited to mitigate this so-called hard trade-off? Okay, so with that, I'd like to thank you all for listening. I must thank my mentor, Eve Marder. She's fantastic, and she's very good at trying to get sense out of me. And I hope you got some sense out of me today. To my PhD supervisors, my collaborators, to funding, and of course, to you all for listening, and for the search committee for inviting me. Thank you.
In something like the stomatic gastric ganglion, how do you think the individual cells know what their burst cycle should be? Because it has to be right, or the stomach doesn't
Right.
work.
The line we take here in this model framework is that they don't. They use some surrogate. They use some surrogate feedback variable, and over an evolutionary timescale, that surrogate feedback variable turns out to be appropriate for maintaining the physiological variable. And then, of course, when this thing is hit by some unexpected perturbation, it doesn't always fix the problem appropriately. So, yeah. Yeah.
In your presentation on the ideas underlying neuromodulation, you presented the blindness of the homeostatic mechanisms as a virtue.
Yes.
Right, but of course it's also a weakness, right?
Absolutely.
I mean, when you're just regulating optimum average calcium, obviously you can have bursting patterns, you can have spiking patterns.
Yes.
So how do you achieve a target of a bursting pattern as opposed to a spiking pattern? With that, and I think you said that particular sets of regulatory variables will ensure that you get one of those?
They do. So the way that it works is that you have this cloud of initial conditions in this model, and you can think of this like progenitor neurons, and they sort of grow and differentiate. And they're using this dumb feedback signal to get where they need to go. And under ordinary developmental circumstances, this works very nicely, and they move to some region of parameter space where things work. And what I'm saying is that if you push these things as they're growing, they may or may not get there. And if we allow them to get there and then acutely push them to another space where they can't sense this discrepancy, then one of two things can happen. It can either be a disaster and completely pathological, or it could actually open the avenue for modulation. So there's a sort of trade-off here. The flexibility of the rule and the way that it allows the neurons, in a sense, to have several constraints. And one of them I showed was temperature robustness, for example. The other one might be the ability to neuromodulate. The cost of that flexibility is that they're not necessarily going to be able to sense all aspects of their activity and compensate for all kinds of perturbations. But it's a completely open question as to where that trade-off sits and how tightly tuned certain neurons are and how flexible other kinds of neurons are. And I think that's going to depend on what kinds of neurons they are, what job they do. Does that make sense?
Yeah, it does, although I still wonder how you sort of find your way to one part of parameter space.
Right.
I mean, I guess you said, as you say, there's a trade-off between always targeting one pattern and being very flexible.
Okay.
Do you think there's a role or a need for multiple calcium sensors in?
That's one possibility, absolutely. So we're not ruling out other sensors. Clearly, there's likely to be others. What we're saying is that the number of sensors is likely to be far less than the number of components that are regulated. Anytime you have that situation, all of the kinds of things I've talked about can happen. You can get aberrant compensation, you can have a certain amount of flexibility, and of course it permits variability. And because we see that, I take that as evidence that there is actually, that the system is underactuated in a sense. Okay, there's a question.
I just want to follow up on Gary's question. So whether or not it's the calcium sensors or some downstream elements that are being regulated differently in different cells. So in basket cells and cortex, you have maybe 10 spikes per second in an active state, but the amount of calcium per spike seems to be lower than in pyramidal cells where you might have 1 spike per second in an active state. And this might bring evoke calcium into a fixed range across cell types, which would require you to have fewer sensors.
Yes, yeah. So that's one way of thinking about it. The other thing is that in this model, of course, we're quite foggy about where this calcium influx lives. In the simple-minded model, we're assuming this is something like nuclear calcium that can activate certain things like Krebs and so on. And what you're describing is a situation where you have cytosolic fluctuations in calcium. And of course, there's any number of ways of buffering and controlling and transducing that signal from cytosol to nucleus. So yeah, that's one way that it can work. And the prediction is that if you change the calcium buffering system in a cell type, then you may get some sort of compensatory response in this cell and unmask how it likes to regulate its conductances. And these are the kind of experimental manipulations that I'd like to do in a reduced system like the neural cultures I was describing. Yeah.
So in the simplified model of 3 conductances, you showed that it was possible for one of them to have sort of the opposite than expected relationship. In the full model of everything that sort of works best for everything, are there cases where the
Absolutely.
relationship is switched?
If you want. Yes. So that can certainly happen. And in the more complex model, in the end, locally, it behaves like the simple model. So when you just have this one sensor and multiple different types of conductances, 50% of the time you're going to go in the right direction. So yeah, the flexibility is there as well.
I guess what I'm thinking there, how much tinkering did it take you? I mean, presumably...
Very little. And that's why is that so the insight came from just working with a simplified set of equations. And I said, well, I'm not going to have these sodium channels and calcium channels and so on. I'm just going to have leak channels, essentially. And once I realized it was easy there, I just said, well, what's going on here is timescale separation. The regulatory control mechanism is averaging out, smoothing out the calcium signal. So as far as it's concerned, all of these different ion channel types behave kind of like a leak. So if it's easy to solve it in the very simple case, it should be easy to solve it in the complex case, and it turned out that it was. We can throw in almost ad hoc, 50% of the time ad hoc regulation parameters, and this model converges and works fine. Yeah. The other advantage is that you could actually add on something else. So you could have a synaptic plasticity rule on top of this, for example. And of course, all the other constraints I described, such as temperature, robustness, modulation, and so on. As you sort of fill up the available space, these things will start fighting. And I think that's just a very interesting theoretical question to address as well, is when that happens, how it happens, and whether that's seen experimentally.
We take one more question?
Sure.
One more question? Okay, let's thank the speaker one more time.